Good afternoon, everyone. I'm Matthias Blum. I'm working at something's missing. I'm working at MBLBI in the InterPro and PFAM team, and today I'm excited to talk to you to present InterProScan 6, or new implementation of our pipeline for protein function annotation. For those of you who don't know about InterProScan, that's fine. It's no one's perfect. So InterPro is a resource that functionally analyzes protein sequences to classify them into protein families, but also detects the protein domains and functionally important sites. It does this by gathering predictive models from different well-known databases, CAF, PFAM, and CDD, etc., etc. And InterPro curators are integrating these protein predictive models that we call signatures into InterPro entries, and they add functional annotations, reference to the literature, Go terms, and so on. On the other side, InterProScan is a software that lets you scan those signatures or predictive models against your sequence. So basically, it allows you to functionally characterize your sequence. It doesn't just report which signature hits which sequence, it also reports the functional annotation that were added by InterPro curators. It's widely used in genome annotation projects, usually often as a first pass to do functional annotations, and it's also a core component of large-scale projects that are at MBLBI, including UniProt, Ensemble, and Magnify. In the beginning, Amos created 3Sprot, and everyone saw it was good. So 3Sprot is this reviewed portion of UniProt, so it's a database of high-quality, manually created sequences. But in the 90s, as sequencing project really took off, it became impossible to manually annotate and characterize all sequences. So this is why Tremble was created. Tremble is the unreviewed part of UniProt, and it contains automatically annotated sequence. So the idea was to have this collection of unreviewed sequences, but with an automatic annotation made available to the community as fast as possible, and eventually they would be included into 3Sprots. At that time, also, you had new projects that were created, such as PFAM, SMART, and so on. So these projects were created to transfer the knowledge from well-characterized sequences, usually in 3Sprot, to new uncharacterized sequences in Tremble. And because those databases, again, PFAM, SMART, CAF, etc., they often describe the same protein families and the same domains, the InterPro consortium was created to remove redundancy, to build on the focus and the strength of each database, and to provide kind of a one-stop shop for protein classification. 4 years after the creation of Tremble, Tremble is already 3 times the size of 3Sprot. It's over 400 times bigger now, so it's unlikely that Tremble's sequence are going to make their way to 3Sprots. But that shows that scalability is really a major challenge when annotating sequences. And InterProScan, which is the current implementation of our pipeline InterProScan, has served well the community over a decade, but the code is 12 years old. So it's difficult to maintain. It's nearly impossible to add components, especially when those components have conflicting dependencies. So we decided it was a good time to start from the ground up and redesign everything. So for the aim of InterProScan 6 was to address all the limitations we saw in InterProScan 5. So we wanted a system that was scalable, that could run everywhere, that basically is on your laptop, on the HPC cluster of Institute, in the cloud. We wanted something that was fully reproducible, that used containers, so you don't have to worry about dependencies and installation. We wanted something that was modular, so maybe that's more for us, but something so it would be easy to add components in the future, especially in those exciting times when you have a lot of new deep learning models that appears and they become available to the scientific community. We wanted a very simple installation. We didn't want users to have to download an archive, extract it, run an install script, edit configuration file. We wanted something that works out of the box. And again, we wanted to have something that was easy for us to maintain. So we can kind of provide support for the next decade of sequencing and protein classification. So we choose Nextflow as the backbone of InterProScan 6, which makes it very easy to scale data, to run or pipeline anywhere, local, HPC, cloud, etc., etc. Each pipeline steps runs an independent process, and they are their own containers. So again, no worries about dependencies. And it's very easy to migrate your workflow or your process from InterProScan 5 to InterProScan 6, because the input is still a FASTA file of your DNA or protein sequences. The command line options are roughly the same, and the output format are the same. I don't know why I wrote JSON and TSV, that's not true. All the output formats that are supported in InterProScan 5 are supported in InterProScan 6. Another key architectural change that we did in InterProScan 6 is that we decoupled the data from the software. So InterPro is released roughly every 2 months. So if you want to use the latest data of the latest version of InterPro, because you want to have access to the latest Go terms added by curators, you want to access the latest annotation added by curators, also because you want to add the latest version of PFAM, CDD, Panther, etc., etc., you have to download InterProScan, which includes the software, the InterPro data, and the data for all the member database. Doesn't matter if you use it or not, you have to install it. And if you always want to have the latest version of InterProScan, but you also want to keep old version because so you want to be able to reproduce the runs of 1 year ago, because someone is going to ask to do something when you submit your paper, well, you need more and more disk space. So this is something we changed in InterProScan 6. The software and the data are handled differently. On runtime, you just do -- InterPro and the version of InterPro you want. If you only want to scan your sequences with PFAM, or only the PFAM data file is going to be needed, and it's going to be downloaded automatically. You don't need to download anything. So that saves time, disk space, and it's just easier to run. Of course, all the tools analysis that are in InterProScan 5 are also supported in InterProScan 6. But because we're re-implementing everything from scratch, we thought it would be a good opportunity to improve the performance for stuff that were not possible before. And I have 2 examples here. So the first one is CAF. You probably know that CAF's superfamily are subdivided in functional families called FAMs. And in InterProScan 5 and 6, you can basically annotate your sequence with both gene 3, CAF gene 3, so basically superfamilies, and with FAMs. But because we don't want to scan all FAMs, because there's a lot, what we do is we do a pre-filter with superfamilies. So we only scan FAMs whose superfamily was already identified. In InterProScan, it's not possible to just identify the superfamilies once for the final output file and once for the pre-filtering default FAM. It's not possible. We have to run it twice. In InterProScan 6, that was solved. So on the top right, you have a chart. Okay, you should be able to see it. That shows the runtime to annotate the human proteome with CAF gene 3 and CAF FAM in InterProScan 5 in orange, and InterProScan 6 in blue. And you can see in blue it's faster. Also, something we did is optimize SMART. So SMART, for those who know, still uses a very old version of Hammer that is used to detect domain in sequences, and it uses Hammer 2, which is, well, very inefficient compared to Hammer 3. So in InterProScan 6, in order to not scan all SMART models with Hammer 2, we do a pre-filtering again with Hammer 3, which is very efficient, and we only do a second pass with Hammer 2 for models that were found with Hammer 3. Because InterProScan 6 is way more modular than InterProScan 5, we can finally start to add tools that have more dependencies, that are more difficult to install. And I have 2 examples here. So in InterProScan 5, you have single P4 and TMHMM2. Single P4 is used to identify single peptides, and TMHMM2 is used to predict transmembrane helices. So those 2 software, single P4 and TMMH2, one is almost 15 years old, the other one is over 20 years old, and their author recently published alternative based on deep learning that provide a better accuracy. So we replace those legacy software by the new one, single P6 and deep TMHMM. Of course, there are deep learning methods, so they're a bit heavier to run. Same charts, human proteome, InterProScan 5 using the old version in orange, and using CPU in blue. So of course for single P it's slower, but if you use GPU in green, it's much faster. Unfortunately for TMHMM, only run the GPU, it's already quite slow. I don't even want to imagine what the CPU looks like. But that shows that basically InterProScan 6 is future-proof. We can add deep learning model and they run. And about the future, one of the most exciting future thing we're going to add to InterProScan is InterProN. So InterProN is a deep learning model developed by Google DeepMind. It's trained on all InterPro data, and it's very, very good at detecting matches that are missing, that traditional methods fail to detect. So usually traditional methods are ISMB or BLAST matrices. And here you have a simple chart that shows the sequence coverage of few model organisms or key species. In blue, in dark blue, you have the sequence coverage found with InterPro traditional methods, and in orange, you have the additional coverage you get, you achieve when using InterPro. So that's a very exciting project developed by DeepMind. InterPro predictions are already available on the InterPro website. They are available through the REST API and to download for the EBI FTP. But we plan to add the deep learning model in the future InterProScan so you can annotate your own novel sequence with this method. Of course, we want to make sure that InterProScan 6 results are highly consistent with InterProScan 5. So what we did, we annotated all the sequence in SwissProt, which is roughly 500,000 sequences with InterProScan 5 and InterProScan 6, and we looked for each analysis in both pipelines, the precision and recall. So just for your FYI, the Y chart started 0.99, so it's pretty good. We do have some discrepancy where we hopelessly hunting, but yeah, there's a pretty good concordance between InterProScan 5 and InterProScan 6. Something else that's quite important, again, it's performance. I already showed you a few charts that show the difference in performance between InterProScan 5 and InterProScan 6. But what we also looked, it's the overall runtime when running the full pipeline. So not just one analysis such as PFAM or CDD, but all those core analyses, except the deep learning method, of course, for a few reference proteomes. And overall, we see that InterProScan 6 is faster. Something else that's quite important in terms of performance is what we call the match lookup. So in InterPro, we annotate all the sequences unipark. So if someone somewhere tries to annotate a sequence that turns out is already in unipark, we have the result for that. So there's no need for them to run that InterProScan locally. I mean, they can run InterProScan locally, but they shouldn't have to run all the underlying tool that's going to annotate the sequence. They can just fetch the results from InterPro. And that already existed in InterProScan 5. And we call that the match lookup service. We completely re-implemented from scratch again in InterProScan 6, and now we call that the matches API, and it's much faster. Just as an example, if you take the Homo sapiens proteome, so over 20,000 sequences, if you disable this match lookup, so you just run everything locally, InterProScan 5 takes about 9 hours, InterProScan 6 takes about a bit less than 7 hours. Now you turn on this match lookup. Well, InterProScan 5 is going to be faster, but it's still going to be quite slow. It's going to take 4 hours to basically just retrieve pre-calculated sequence from the match lookup. While InterProScan 6, yeah, everything's on your computer in less than 20 minutes. And an issue with InterProScan 5 is what the match lookup, the old match lookup was really, really designed for InterProScan 5 and nothing else. It has multiple endpoints. You have an example of one of the response, and it's a weird, clunky XML with weird record as encoded as CSV. It's really impossible for developers to benefit this outside of InterProScan 5. So yeah, I mean, InterProScan 5 is faster, but yeah, that's not very good for the community. While for the InterProScan 6, the match API, there's one single endpoint. You can submit one or multiple MD5 that represent your sequences, and the response format is in JSON, which is also the exact same structure that the JSON generated by InterProScan 6. And that was really a huge missed opportunity that the old match lookup service was not more developer friendly, because we have nearly 1 billion sequence in Unipark, which corresponds to about 30 billion InterPro annotations. So we had this goldmine of data that was kind of hidden behind a difficult to use API. So we really hope that with the matches API, we just don't increase the speed at which InterProScan can retrieve those information, but we can also encourage the community to reuse this data, whether it's in their workflow or if they have a website and they want to integrate some InterPro annotation in their websites. In summary, InterProScan 6 is not a simple update or upgrade for InterProScan 5. It's a complete re-implementation of a software that was widely used in the community for over a decade. It's reproducible, it's modular, it's continuous, it's very, very easy to deploy and to run. I show you it's faster than InterProScan 5 with a very high consistency of annotation between InterProScan 5 and InterProScan 6. And yeah, it enables integration of new tools and methods like we couldn't do that with this InterProScan 5. So it's really future-proof, and yeah, it will be able to support the next decade of advance in protein classification. In terms of roadmap, we already have a beta that was released a couple of weeks ago, and we aim to have a stable release in September. We'll still maintain support and release InterProScan 5 for a couple of more months, but if you use InterProScan 5 in your research or in your service, I really encourage you to test InterProScan 6 and let us know what works or what doesn't. And with this, I would like to thank the team behind InterProScan 6, especially Lays and Emma, who worked a lot on the pipeline itself, and also Alessandro, who's working on the integration of InterProScan 6 results in the website. You can find InterProScan 6, the GitHub repo on just a Google search, or you have a short link here. And if you don't feel like chatting with me during the conference, feel free to reach out online. And with this, thank you for your attention, and I'm happy to take any question, or do we take a question later?
Thanks very much for this. And we have a first question here already.
So great talk. I was a PhD student at the EBI when InterPro started. And I never thought that you guys will be running it on a language we developed 10 years later, because we developed Nextflow and we never thought this would run this way. Now, my question, we are now toying with the idea of defining modules as classes in Nextflow, as input, output, and operation. And that's something very hot on our plates. And I'd like to know your thought about this. Will this fit in your vision of this InterPro 6?
It would. I mean, to be honest, I see your name. Yeah, your Nextflow authors. I'm not going to blame or bash Nextflow, but it was sometimes difficult to run it because we don't have like just container that already existing in biocontainer. We had to run to write a lot of our code ourselves. If you change the way that processes interact, input and output, we may have to rewrite a lot of our stuff.
No, we will not change anything.
Okay.
We will simply define modules as classes. And
then it's fine.
They are not defined as classes these days. You know, you have to, if you want to do the multiple alignments, you have to pick them and you have to add them one by one. But if they were defined as classes, you will know that all of these guys have the same input, the same output, and do the same operation. So for instance, all of your profile or motif finders effectively belong to the same class, where they add a layer of annotation onto the sequence with same output and same input. And I'm simply saying that we name these things, we define these things in a consistent way so that they will be a class. And programmatically, you could expand InterPro 6 with whatever has been produced that fits the definition.
Then yes, I would be interested to, it's still a bit blur how it will work, but yeah, I would be interested to see that.
Okay.
Yeah, thank you. You were mentioning the fact that InterProScan 6 works a lot faster than InterProScan 5. Now, when you dive into the details of this performance increase, is it coming from the use of Nextflow? Is it coming from the use of newer versions of software that you call?
So we try to not change the version of the software. For instance, one of the core components of InterProScan 5 is Hammer. It's how you annotate your sequence. And we use the same version because otherwise it will not be reproducible. The main advantage we see in the performance is not in the process, in the binary executable itself. You run Hammer 3, InterProScan 6 or InterProScan 5, it's more or less the same speed because same input, same options. Where it really changed, it's the way the post-processing, so reading the output file, it was done in a clunky way in InterProScan 5, even though it was Java. We were implementing most of these functions or process in Groovy, which is also based on Java, but yeah, in a better way. And also InterProScan 5 has this kind of internal database, and multiple thread has to, when you use multi-threading, they have to kind of to wait, and you lose a lot of time there. So they started to, the authors of InterProScan 5 did this multi-threading to well, make things faster, but there's a glass ceiling, and there's a plateau. And with InterProScan 6, since almost everything is using files, you don't have this internal database, so it's just faster.
Okay, thanks.
Thanks very much. Applause again for the speaker.